Hello everyone, this is Xuexue Liu from CGT in France. Welcome to mapDATAge. The mapDATAge application is a Shiny R package to charge HMD data through space and time. It takes simple text files as input and contains different panels to draw a number of maps and plots that are common to most HMD studies. It includes 9 panels. They are part of the welcome menu. SMAP, AMAP, trajectory, ANCESTRY, PC, MULTIPLESNPS, HAPLO, and finally, ONECLICK. In this video, I will make you familiar with the 9 panels. Our code is open and available through Github. Before using mapDATAge, you need to install the R dependencies, and this can be done by running this command. In case you don't want to install mapDATAge locally, you can run any of the 2 commands in your computer. This will allow you to avoid local installation. We, however, strongly recommend that you install mapDATAge locally, and this can be done through here. For example, you download the code here in this folder, and then unzip it. Finally, run this command in R or RStudio. mapDATAge needs some data for preparing plot and analysis. You can import your data by using the browser. And click on the Run mapDATAge button. If your data file is not correctly formatted, an error will be flagged. Here we don't have formatting issues. Please verify the input file. Data can be further sorted and searched. For example, click on the sex, and the data can be automatically sorted. It can also search for any of your requests. For example, then is this the detailed information of this sample. Now let's move to the SMAP panel. This map panel includes a sum panel and a main panel. The sum map panel provides parameters for visualization, including colors, sex, species, time, geographic range, and more. And the main panel shows the map and graphs. Click on any of the data points. It will show the list of metadata associated. Here we can see all the samples from the same set, as requested by the label selection parameter. Of course, additional labels can be displayed, such as site, species, sex, and all. Then click on any of the points. The selected information was displayed here. You can also scroll up and down to zoom in and out the map, or use the tools on the left. For example, polygon and square symbol to select manually a region of interest. The temporal distribution of the selected samples will be displayed on the bottom. You can always go back to the original map by using the Reset button. You can or clear the selections. Finally, the mini-map on the top corner, it shows the region that is considered. It can also be hidden by click on the small arrow. Next, AMAP panel. This provides the geographic and temporal distribution of the 2 allelic states at a given locus. The example data is the mutation of GSDMC in horses. The left panel is similar with SMAP. We also introduce SMP filters if your input file includes more than 1 SNPs, and select allele type. This is necessary. Please select genotype or read count option according to the type of the data provided. Chart type, allowing users to represent frequencies through pie chart, bar plot, polar area, and polar radius at the main panel. And in the main panel, it shows the distribution. The chart will be automatically merged for those samples merging the same GPS coordinate. You can also group neighbor samples from nearby locations and input the grid size here. For example, you put 2. It will group all the samples within the grid of 2 together. If you want to save your output, please select your time bins, for example, 1000 to 5000, and a slice of 1000. Then set the output path by providing a project route. Once clicking on the Draw Temporal Maps button, it will automatically generate HTML output files delivering the corresponding maps in your output folder. Then it will generate here. Now the AMAP panel is finished. Let's move to the trajectory panel. We used the SDMC mutation of C allele as an example. This panel can draw the allele temporal trajectory at any given locus present in the input field. The left side of the panel provides the same control parameters as those provided in the AMAP panel. This is the geographic distribution of C allele with clustered circle markers labeled according to the number of samples carrying present in the given locus. Click on such markers will unfold all underlying samples and associated metadata. The blue, green, yellow, and orange colors reflect the number of individuals. The window size gives the number of years considered in each time slice, while the step size gives the number of years considered for shifting one time slice to another. Once the allele type is selected, for example read counts and sample times 100, here it draws the temporal distribution of C allele by time from 0 to more than 5,000 years ago. Within 1,000 years window size and 500 years step size. The blue line is the allele frequency estimate, and orange dots indicate the number of samples considered within each time bin. You can obtain more information by scrolling your mouse over each estimate. Now it's time to the Ancestry panel. This panel projects individual ancestry profile on a map. This feature allows users to immediately explore possible changes in the geographic distribution of ancestry profiles. Please import the raw data. It was provided by mapDATAge and it was taken from Librata 2021 paper. Here we provide the time point, which allow you to join two maps, one before and another after the selected. Similarly, if you want to draw maps of time sliced on a given ancestral years, you can use the time slice parameter, for example 1000 years. It will tell the mapDATAge to draw ancestry components every each 1000 years and save this result in the folder you specified here. Now you can take advantage of the grid parameter to merge sites within neighbor archaeological sites, for example 2. It will be visualized on the right. After Ancestry panel, we go to the PC panel. This panel allows users to visualize the genetic structure of their datasets using PC, MDS, or any multivariate analysis result. Please import your original file here, and also this file. Then the PC plot can be showed automatically. The data was the same with previous panel. The geographic and temporal distribution of the samples to be visualized below. Here is the time for the multiple SNPs panel. This panel helps users to map the frequency of specific alleles at one or multiple loci. Please multiple choose your alleles of interest. Here, list of genomic locals driving coat color variations in horses, and display with bar plots on the right. Each bar plot provides allele frequency of each selected locus. Second to last, HAPLO panel. This panel helps users visualize the geographic distribution of mapDATage or Y chromosome haplogroups within a pre-selected time range. It can also be used to map any quantitative category of data. Here, we will use the example data that mapDATage provided. We show the Y haplogroup in 1,000 European males. The spatial distribution will be shown on the right. You can make use of the grid size parameter and select labels you want to display as shown on the previous panels. For example 2. The last, ONECLICK panel. It allows users to provide a predefined set of parameters to be applied to one data file. This will automatically generate all possible corresponding figures without any need for filling up parameters. Please import the parameter file here and check if it was correct. Then set the output path. Click on this, you can get your result automatically in your folder. That's all. I hope the mapDATage will be useful for you. Please don't hesitate to send me your suggestions for improvement. Bye.